Hello, I'm Laura Cassidy from the American Chemical Society. Welcome to this news briefing from the ACS Fall 2019 National Meeting in San Diego. We're joined today by Dr. Vivek Kumar and Dr. Biplab Sarkar from New Jersey Institute of Technology. They're studying peptide hydrogels that could help heal traumatic brain injuries. Dr. Kumar?
Hi. So it's actually really interesting work that our team at the New Jersey Institute of Technology has been working on for a couple of years now. This work was led by Biplab Sarkar, my postdoc there, and some of the unique peptides that we're making from this platform have been translated not just for neurogenic applications, but angiogenic, and a variety of other applications that we've talked about at this conference, in fact.
So our talk, our platform really depends on these peptide hydrogels that are injectable directly into the brain. And one of the problems that many people have after they have brain injury is that they have issues walking, they have debilitating conditions that can last not only minutes or hours, sometimes up to days or even weeks, and so on. Not only professional athletes and boxers have these problems. In fact, most of the traumatic brain injuries happen in mostly older folks who fall and sustain an injury. And one of the issues that we are trying to solve is how do we develop a material that can be injected directly into the brain, into the injury site, that can stop some of these processes, these pathological processes, from progressing. And in our presentation, in the first slide, we show these setup that we test our materials in. We basically use a rat model, and the specific model that we use is a hammer to induce an effect of traumatic brain injury. And after injecting our materials, we test in the untreated animal and the treated animal how well our device actually works. So in the next slide, we can see on the left are these nanofibers. They are very small nanofibers, so like so about 1,000 times smaller than the width of your hair, for example. And these nanofibers form these gel-like structure when they are in solution. And what they can help is that they can help these neurons on your right, these are cortical neurons from rats, to hold on to these nanofibers and survive longer term. And one of the issues that we really try to solve is that after traumatic brain injury, one of the issues that happens is that we have lots of glutamate ions, which is the same as MSG, but doesn't come from the food, obviously. And that glutamate ion really kills lots of neurons. And this is called glutamate-dependent neurotoxicity, excitotoxicity. And we showed in vitro that our hydrogels can somehow capture, slow down that excitotoxic process. And we showed that we could translate to some extent, at least in the rat model, this is obviously not in humans yet, that we can use that model from in vitro to in vivo applications up to 7 days. We have not done any long-term studies about 30 days or up to maybe 6 months. That would be the ideal scenario. One drawback that I would like to point out to you is that our study, apart from being done in rats, it's also is done injected directly in the brain, which is better than opening up the skull and putting a material there, but it's not ideal. Ideally, you'd like a pop-up pill or at least do an IV, intravenous injection. So we're trying to solve that problem. How do we inject this material in the bloodstream and pass the blood-brain barrier? So that's like this barrier between the blood and the brain tissue to actually supply the material in the injury site.
Very good. All right, are there any questions? Please state your name and affiliation before asking your question.
Hi, my name is So Young from UCSD, and my question is, what kind of peptide are you using to form these hydrogels? And the second question is, do you expect these peptide hydrogels to cause immunogenicity?
Right. So it's really interesting. So this platform that we've developed or continued developing from some of the early work from Jeffrey Hartgerang back at Rice University is based on a short 12 to 20 amino acid sequence. They have terminal charged charged amino acids with hydrophilic and hydrophobic mid-blocks, alternating hydrophilic and hydrophobic amino acid residues. We call these self-assembling peptides or multi-domain peptides. And in general, we've shown that they're pretty well tolerated by the body, so we can inject them subcutaneous, intramuscular, a variety of different tissue sites, and we don't really see a significant amount of inflammation, be it acute or chronic. We do know that our materials do persist in tissue spaces. Some of the work that Biplop presented earlier on, and I guess we don't have histology here, but we show that we have very good cellular infiltration to these materials, and the degradation is somewhat mediated by immune cells, be it phagocytic or enzymatic degradation. That's something we're still looking
Just to
into.
add on that, because when people hear immune cells, they hear B cells or T cells. These are macrophages mostly, which are normal in these kinds of materials.
Right, and in addition to that, that's a great point. In addition to that, we don't really see a prolonged immune response or any overt signs of inflammation as these materials degrade over 2 week, 3 week, or longer periods. And I guess the last part to add to that is we have actually extended this platform from mice to, or rather tested this platform in multiple tissue sites in mice, rats, and even dogs now. So, yeah. Thank you.
Hi, so it's Kath O'Driscoll from Chemistry and Industry magazine. I'm wondering, I know a lot of the issues to do, because there's a few emerging therapies coming through for brain injury. And I understand that one of the issues is how soon the compounds have to be administered after the injury occurs. Could you say something about that?
So I think Biplop touched on this just now, so maybe I'll let him continue some of those thoughts on how we can get this to the site of injury.
Yeah, one of the issues, exactly what you said, like it's very difficult to find a patient after seconds after they have suffered an injury. The ideal scenario would be they would go to the ER, get an injection, either IV or brain. What we have tested is immediate, the gel was injected immediately, within like seconds after the injury. So that's somewhere we would like to go. We're not there yet.
So, and extending that, if we can figure out, so for example, when someone has a stroke, you want to administer tissue plasminogen, TPA, within 30 minutes or something like that. So if we can find a sweet spot at which point our therapeutic is still effective, either deliver intravenously, orally would be amazing, but we doubt that'll happen.
Ideally, you would want
transcranially.
these to be administered by paramedics. So it would be somewhere you can store in an ambulance that they can inject it. One of the advantage of these materials is that they're not cell-based, right? So they're acellular, so that you can keep them outside for a long period of time, over 3 months, 6 months. They have good shelf life as well as low batch-to-batch variability, which is very important for these kinds of quick applications.
And finally, this is the place to talk about chemistry. You know, it's not often that you get a chance to do that. So one of the nice things about these materials is that they do self-assemble into these hydrogels, self-assemble into beta sheets, and then subsequently entangle into very well-hydrated hydrogels. While we haven't done formal stability studies, either in vitro or in vivo, that is something that we're pursuing for a variety of different peptides in this platform. But we do think they have very good stability at a range of temperatures.
And in the lab, we keep them at 4 degrees Celsius, which is, I think, obviously limits to some extent, because we have not tried to test them, keep them at the room temperature to see how they degrade over time. But we know that 4 degrees Celsius, they last for months, years.
Thank you.
Sophie Rovner with the American Chemical Society. Can you tell us a little bit more about what's happening in the brain after you inject these and how they interact with the neurons?
So what happens in the seconds after you have a brain injury is that your axons, the neuron, the feet of the neuron, they get cut off. They get axonal tear. The microtubules get disrupted. So that's like more like a physical injury or a mechanical injury. That happens super quickly. And you get some bleeding in your brain. What happens after minutes to hours and beyond, this is called a secondary injury, which is a lot of immune reaction in a way that because brain is not supposed to have these foreign cells or the blood-brain barrier disruption is one of the problem. Lots of glutamate gets released in the brain, causing a spike of calcium ions in neurons that kills them. You also have something called chronic traumatic encephalopathy, which is basically somewhat Alzheimer's-like phenotype, which happens over a long period of time. So they have seen in boxers after decades after they have left the sports. So the brain's mechanism of clearing out debris, or how the nutrients get into neurons and how the toxic chemicals get out of neurons, both processes are affected. And we think that if you can vascularize the brain more effectively in the short term, that will solve that problem so that brain doesn't go through this, what is called energy crisis. So basically, it doesn't have enough ATP to run the machine.
So to expand on that to the entire platform, we've seen that while this peptide that we've created is pro-neuroprotective, and if you inject that, you have a certain degree of vasculature and neuroprotection of the neurons in the vicinity. More broadly, we see in this platform, this hydrogel, is that you have a lot of cells infiltrate these scaffolds, and at a constant level, a basal level, you have some level of angiogenesis or tissue regeneration or tissue formation. So exploiting that along with the persistent presentation of a signal. So one of the other aspects of our self-assembling hydrogel is that you can syringe aspirate, inject it, and it stays where you put it. Some of the work that Biplab presented earlier, that I will show later on today, talks about how we can take these materials, inject them into local tissue sites, and modulate those environments. And depending on the signal we're presenting, be it neurogenic, angiogenic, or what have you, we can tailor those environments specifically to those different phenotypes.
To add on to that, generally when you inject a drug, sometimes it gets cleared very fast because through your kidney, for example. What we can do, because these materials are themselves the active material, we're not putting any secondary small molecule drug, any molecular antibodies in there. The material itself is the drug. And that gives us an opportunity to keep them in the active area for a much longer time because it's a solid material. It's not a solution that goes through tissue clearance super quickly.
All right, sort of a far out question here. You're talking about neurogenesis, or?
Go ahead.
Yeah. So for the current work is mainly preventing neuronal death. So neuroprotection. At this point, we're not ready to perhaps talk about the neurogenesis aspect because another scenario where you can what you can think of is recruiting neuroprogenitor cells from subventricular zone into these nanofibers through some sort of chemokinic kind-like signals so that create new neurons and synaptic connections. We are not ready to talk about that yet.
Right, and
Okay.
yeah.
All right, fair enough, because I was going to ask if you could make someone smarter.
Hi, so it's Kath O'Driscoll from Chemistry and Industry. I'm sorry if you already touched on this, so I was a bit late. But did you say anything about ependymin, this protein sequence that you're using? Is that a natural protein, or what is that, and how does it work?
Yeah, it's a natural protein, and the ependymin mimic that we are going to use, actually activates a neurotrophic factor-like pathway. There's a neurotrophic factor called BDNF, brain-derived neurotrophic factor, and it activates a protein intracellularly called AP1, activating protein 1. And that's generally how the ependymin mimic works. What we can say at this point is that we don't have any proof at this point that we can do neurogenesis with these materials that we presented, but that's actually a very promising next step.
Okay, thank you.
Bela Buslik, ACS. From the picture that I see there, those nanofibers seem to be fairly sizable. I presume they're bundles of peptides. And how do you distribute them at the right place, because when you take a bunch of sticks, put it in there, well, they'll stay there for a while. You expect the brain or whatever, wherever tissue you're injecting it, I mean, to do something to disperse it, I think.
Right. So unfortunately, there is no perfect imaging technique or enough space on slides to show all of it. But if you do make it down for my talk later on today, these hydrogels that form have a three-dimensional structure. So they're a three-dimensional, kind of like a sponge, meshwork. And they're nanofibrous. So those dimensions that you see there in AFM is about the expected dimensions of an individual peptide on its width. So those individual peptides come together to form beta sheets, and they further aggregate into long fibers. Those fibers then entangle. So the width of the peptide is about what you expect there, about 10 nanometers. And when we measure the height, it's about 1 to 2 nanometers. So two of those peptides stacking on top of each other. Now, in three-dimensional structure, we see that these fibers actually extend into very long, infinitely long, aspect ratio-wise, fibers that entangle and solvate, hold a lot of water, so they form very nice hydrogels. Since they are held together by non-covalent interactions, they break very easily, reform very easily, so we can syringe aspirate, inject, and right after we inject these hydrogels, they re-self-assemble. They form these beta sheets almost instantaneously, and they stay persistent in the tissue. So over time, we see a variety of different cells infiltrate these scaffolds, biodegrade these scaffolds, and clear them either through phagocytosis or enzymatic degradation or something like that over month-long periods.
And what you're getting once they're in place and do their function, they eventually get dissolved by the body and
right. So one of the things that I personally, or our lab is very interested in right now, is trying to figure out how we can track these. So if you put a big, bulky fluorophore in these short peptides, you're going to interfere with their self-assembly. It's just not going to self-assemble the same way as a native peptide, and might interfere with activity, binding to cell receptors. One of the things we're interested in doing now is maybe radioactive labeling these peptides or something like that, and then tracking their biodistribution, and also trying to understand exact binding.
And these peptides are relatively low immunoactive.
So we've done for this platform, we've done primer blasts in silico. We've also injected these materials for long periods, so they degrade over month-long periods. Typically, one would expect to see an adaptive immune response, usually around the first 2 weeks or so. And we just see that these boluses get smaller and smaller. And when we do histology, instead of seeing a fibrous capsule that walls off the implant, preventing cells from going in there, what we see is, if anything, a very thin or no fibrous capsule, excellent cell infiltration, several hundred microns or millimeters into these scaffolds. And depending on the signal, like Biplab was mentioning, depending on the signal that we present, we can get either a variety of different phenotypes.
Thank you.
Thank you.
I have a question. So these rats with traumatic brain injuries, did they show any behavioral abnormalities, and did these get better with injection of your peptide?
So they are less ambulatory after you have a brain injury, which kind of is expected. We do have some behavioral data, but this is so preliminary. I think it's a stretch to say that we show behavioral progress. Perhaps in the future we can talk about that.
Okay.
Ongoing work.
Any other questions? All right, thank you. The archived version of this session will soon be posted at bit.ly/ACS 2019 San Diego. Please join us for our next press conference at 2:30 p.m. today on a smartphone-based device for detecting norovirus, the cruise ship microbe.